All right, you guys bio people, we're now on Unit 6, Segment 4, and it's called Mutations. This is the last segment of this unit. Mutation is defined as the change in genetic material, so a change in your DNA or your chromosomes. This can be good, this can be bad, this can actually result in no change at all. So let's talk about the types. When we typically think of, and you don't have to write this slide down, but when we typically think of mutations, we usually think of something bad. You know, like something like this frog has the extra leg. You know, obviously there were some mutations that occurred. This frog is not normal, something happened. Could be created in a lab, could be a nuclear radiation, whatever we usually associate with that. While that is true, there's other reasons that can cause mutations. There's something called mutagens. It's a vocabulary word, and it means something that can increase the frequency of mutation in an organism. Mutagens can include chemicals that are in the environment, man or natural. It also can include radiation. Chernobyl is an example of radiation, and that was, there was radiation that spread out over many miles, and children that were born shortly after that meltdown of the power plant were showing very large signs of mutations that occurred. Carcinogen is any substance that actually has a tendency to produce cancer. So this would be like asbestos, DDT, tobacco, those kinds of things that you've probably heard of the word carcinogen. And it can cause mutations, which then lead to cancer. So there is a connection with mutations and carcinogens and cancer. Here's an example of lung cancer. You have a healthy lung here on the left, and you have a smoker's lung here on the right. Very big difference. Lots of clogged little airways, and just really bad. You don't have to write this down, but tobacco has over 28 known carcinogens. Can you believe that? Which is why people who use tobacco in any form have an actual 50% higher risk of developing cancer. That's crazy. The effects of chewing tobacco actually causes cancer, could cause cancer in the jawbone, which could result in actual removal of that jawbone. I mean, it's pretty dramatic and crazy. All right, now, down to the nitty-gritty biology of this. So there's two categories of mutations, two general categories. There are gene mutations at the gene level, so a segment of the DNA, and there are mutations at the whole chromosome level. All right, so gene mutations are going to produce changes in a single gene. So the nucleotide sequence in the DNA, all right? Chromosome mutations are going to produce changes that will impact the whole chromosome. They can be a portion of the chromosome or they can be the whole chromosome, but either way you're going to see it at a chromosomal level. So if you're looking at nucleotide sequences, if you're given an example of a mutation and you're given nucleotide sequences, you are looking at a gene mutation. This is a rule of thumb, okay? If instead you're given an example where you're looking at a whole chromosome, this is a little trick, then you know it's chromosomal mutation. So it's a really easy way to know what you're looking at. Now we're going to talk about gene mutations first. Point mutation and frame shift, point mutation is a mutation that is involving only one or just a few nucleotides. Remember what nucleotides are. They include the sugar, the phosphate, and the nitrogen base. So there are three types of point mutation, you guys. So the point mutation is the umbrella vocabulary word that encompasses these three here. Okay? So substitution is when a nucleotide is switched out for another. Insertion is when one has been added in, inserted, a nucleotide. And then deletion is when a nucleotide has actually been removed from the strand. So these two, all right, so if I swapped out an A for a C, if I swap that out, it actually wouldn't impact the groups of threes. They would still say the normal groups of threes. But if I pulled one out, now you've shifted that whole groups of threes codon. You've shifted that. So the same happens when you delete a nucleotide out. When you're removing one, you shift the whole thing. And you'll see this in a second. I'm going to show you some pictures. So because the whole thing is shifting with both insertion and deletion, we have another general name for those two, and they're called frame shift mutations, and they're a type of point mutation. So still under the category of gene mutation, the frame shift I was just mentioning, these are changes that actually shift the whole genetic message by insertion or deletion, okay, of a nucleotide. These have some pretty dramatic changes because from that sequence, which codes for an amino acid on, it's going to be different. So potentially you're making a whole different protein, all right? So the protein that was supposed to be made will not be made or will not be able to do its job, which is pretty bad. All right, here's the same picture that's in your book. So here's an example. We really have to look closely at what we're looking at here. So the last segment, you guys were showing me DNA sequence, right? Transcribed into the mRNA sequence. And so this would be the normal, transcription. All is good in the world. Translated into the amino acid sequence, this is what we have here. But if there's a substitution mutation, for example, that C that was up here in the DNA could actually be replaced with a T. It's substituted for T. Well, what kind of impact would that have? What does that do? Well, it changes the transcription. What should be a G here is now an A. So when you use that bullseye, amino acid diagram to figure out, key out the mRNA, it's now coding for histine. So the ARG was supposed to be the amino acid. Now it's HIS. Now it's a different protein altogether. It's a different amino acid, which leads to a different protein, which could lead to a dysfunction. Okay? That is called substitution. It's substituting one nucleotide for another. Notice how the threes, substitution only impacted this one. The rest of the amino acids are the same. So all is good except for this one. So its impact is a little bit limited. Now, with insertion, different story. So we have the same DNA sequence, transcribed into mRNA, translated into the amino acid sequence. But if we insert a T between the C and the A, if we insert that, well, now that whole thing shifts. From this point on, it shifts down. This is supposed to be TAC, but it's TAT, and then the C is now over here in the grouping. So it throws everyone off after the first one right there. So it is not reading the first one right, and it's reading all the rest of them wrong. So these are all different amino. This is a big impact, okay? Insertions and deletions are very impactful. So here's deletion. This is showing a little bit different. Instead of showing the nucleotides, we replaced it with actual words. So it's kind of like an analogy. So it's still stuck in groups of 3. Okay? So if this is the mRNA, this is the code on the mRNA. The fat cat ate the rat. The meaning behind this sentence could be considered protein. Okay, so this is an analogy. Obviously, we're not looking at A, T, C, and G. We're looking at other words. So, but if we pretend for a moment that these letters are nucleotides, the meaning behind the letters is going to create the protein. If we delete out the H, it no longer spells the. And if we chunk it into 3s, look at what we get now. Nonsense. It doesn't read, there's no meaning, and it's a very good analogy to what happens when a protein, when a nucleotide is deleted out of the order, it shifts the whole sequence out of order. I mean, it shifts the whole thing, for example, in this case, to the left. So instead of THE, it's TE. The E goes over here, the F goes over here, and now it just doesn't read. It doesn't function like the protein's supposed to. Okay? So substitution, insertion, and deletion are all called point mutations. Insertion and deletion are called frame shift mutations also because they shift the frame of the reading for the amino acid sequence. They completely shift. From that point on, it's read wrong. can be very dramatic. There is an example of a specific disorder that you're going to need to know about for the test, and actually in class I think we're going to do some reading either in your packet or I will be providing you with the sheet. But it's called sickle cell anemia, and you may have heard of it. But all it is, you guys, it comes down to one nucleotide, okay? So here's, this is just the name of the allele, the gene that codes for sickle cell anemia. And here's your DNA. The DNA for normal is CTC. But for somebody that has sickle cell anemia, instead it's a CAC. So what type of mutation is this, do you see? So CTC, and instead somebody with sickle cell has CAC. Which one? Which point mutation is it? If you answered substitution, you are right, okay? So when a normal person without sickle cell anemia has transcription happening, it's transcribed into mRNA as GAG. But a person with sickle cell anemia has GUG as their mRNA, and that is going to read for the wrong amino acid. GLU is the amino acid for GAG. VAL is GUG. So the wrong one is being read. So a different protein is actually translated when this happens. Hemoglobin, which is the part of your blood cell that actually will accept oxygen, it actually causes a sickle shape to your blood cell. And it's kind of shown here, illustrated here. So you have a normal red blood cell here. With this coating, it is sickle shaped. It's got a point to it. So it does not hold oxygen the way that a normal red blood cell does. It holds less oxygen. So you have less oxygen being delivered to your body, which means less cellular respiration, which means that you're tired quicker. Okay? So, and there's other ramifications too. Okay? This also, because of the sickle shape, can actually cause clogging of the arteries, blood vessels, which can be problematic for sickle cell anemia, people that have that. Okay? Sickle cell. All right, more chromosomal mutations. It looks, I'm betting we're probably going to have 2 videos that are going to have to take place because I'm already at almost 13 minutes and I still have more to go. So, but so we just covered gene mutations. Now we're going to cover chromosomal mutations. These are going to be the ones that are going to actually cover a big chunk of the chromosome or the whole chromosome. So you won't be looking at nucleotide sequences anymore. You're going to look at whole chromosome. So there are 4. Okay? Deletion is when segments or whole chromosomes are deleted. Duplication is when extra segments or whole chromosomes are actually repeated, duplicated. Inversion, when you invert something, you flip it. So a segment of the chromosome is actually flipped in reverse order. And then translocation is when one part of one chromosome pops off and adds to another chromosome. So there's 4. We're going to look at a picture of showing each. So we're not looking at, these letters are not nucleotides, by the way. This right here is the centromere. This is a whole chromosome. This is one of the chromatids, one of the sides of the chromosome. So when you're looking at the chromosome, you know automatically it's a chromosomal mutation. So you got to think of the 4 that there are. Deletion, duplication, inversion, and translocation. So let's try to make sense. I have 1 minute to cover this. We have the normal A, B, C, okay, portions of this chromosome, and D, E, F, that's below the centromere. With deletion, a whole chunk is removed, and in this case, the whole B section was removed. So that is viewable, visible, when you look at the whole chromosome. You can see it's shorter, okay? That's called deletion. Now, when you have your normal chromosome, and instead you have 2 chunks of B, 2 repeats of that, that's called a duplication. So the actual chromosome actually looks longer. Now, the other 2 are a little bit harder to tell just by looking at the size of the chromosome, but here you have the 2 legs, I guess you can, and they cross, and then so it pops off. This right here, this little box right here, pops off, flips over, and reconnects. And then we have translocation, where a portion pops off and adds to another